Hello everybody. Today I'm going to talk to you about viruses, microRNA and host interaction. First, let me introduce to you what microRNAs are. MicroRNAs are small RNA molecules that has recently been discovered and has been found to have several roles. These microRNAs are usually 19 to 24 nucleotides long, and their major role is gene regulation. MicroRNAs were first discovered in C. elegans and has been widely studied. C. elegans has been a widely studied model for learning about microRNA. There has been rapid advance in the discovery of several new microRNAs since the first discovery. Um, Currently, there are several thousands of microRNA sequences present in the MIR base, which is the microRNA database which is online. Some of the roles of these microRNAs are growth, differentiation, regulation of gene expression, stress response, immune activation and inactivation, apoptosis, antiviral defense, and many more. Let us look at the biogenesis of microRNAs. MicroRNAs are most commonly synthesized by the canonical pathway. In this pathway, microRNAs are produced by RNA polymerase II, and during transcription of normal cellular mRNA. Initially, the microRNAs are several hundred base pair long and can contain one or more hairpin loops. Just like any mRNA, these microRNAs are called primary microRNAs, and just like any mRNA, primary microRNAs have a 5' cap and a 3' polyadenylation site. The primary microRNAs are then processed by the endonuclease called ROSA, which is in the nucleus. After processing, the primary microRNAs become pre-microRNA, which is 60 to 80 nucleotide long stem loop structure. The pre-microRNA or the precursor microRNA is then transported from the nucleus to the cytoplasm by a molecule called exportin 5. So this pathway is called the canonical pathway, and the later steps are common for all the pathways. So I'm going to explain to you the other pathways apart from the canonical pathway, and then go to the lower steps. The next another way of making microRNAs is through RNA polymerase III, and as we know, RNA polymerase III makes transfer RNAs, and this type of microRNA production is seen in murine herpes virus 68, MHV68. The primary microRNA consists of a 5' tRNA structure. This is then cleaved by an enzyme called tRNase Z in the nucleus, which produces the precursor microRNA, and the precursor microRNA is again transported to the cytoplasm by exportin 5. And the third way of microRNA production is through splicing of mRNA molecule. The intronic regions that produces a lariat folding refolds to form a pre-microRNA, and this pre-microRNA, which is produced from the intron of the mRNA, are called nitrons, and these mitrons are then transported by exportin 5 again into the cytoplasm. And the last and final way that has been discovered so far is through small nuclear RNA, which is directly transported outside the nucleus, but the mechanism of transport is not known. This is then yeah this is then cleaved by precursor. This pre-microRNA is then processed by another enzyme in the cytoplasm called dicer. Dicer cleaves the 3' stem loop structure and creates a double-stranded microRNA duplex. From this duplex, one strand of the microRNA gets incorporated into a protein complex called the RISC complex, which is RNA-induced silencing complex, and the RNA-induced the other strand of the microRNA gets degraded. So these are the ways by which microRNAs are produced. Next, I will talk about how microRNA can silence or reduce the expression of mRNAs. So microRNAs that are incorporated in the RISC complex binds to the mRNA 3' untranslated region of the mRNA, either with perfect complementarity or imperfect complementarity. If the microRNA binds the mRNA with a perfect complementarity, this leads to mRNA degradation. And if the binding is not perfect, and if you have gaps, then you have a translational repression, that is, the mRNA does not produce proteins, and the mRNA is then transported to a vesicle where it is stored, and this vesicle is called the P-bodies. mRNA degradation by perfect complementarity is usually seen in plants and insects. It has not yet been identified in vertebrates. Inside the P-bodies, the mRNA can either be degraded by the endonucleases present in the P-bodies, or they can be stored and can be transported back to the cytoplasm from the P-bodies when the conditions are favorable. So at the time of infection, along with the viral proteins, viral microRNAs are also expressed, and these microRNAs are used as weapons by the viruses, because microRNAs are non-immunogenic to the host, and a host and also the size of the microRNAs is very small, which makes it easy for the virus to mutate and evolve rapidly. And since the viral genomes are not very large, they cannot encode several different microRNAs to target several different genes. But microRNAs, because of their small sequence, can bind several different mRNAs and inhibit their um, production. Viruses express these viral microRNAs to establish a successful infection and maintain active replication. This table shows a few important virus families and the host which they infect, and the number of microRNAs that have been discovered in each of these viruses are also shown. Uh, it's not important to know this table. It's just for your reference. "25 microRNAs have been discovered in Epstein-Barr virus, and 3 of these microRNAs are encoded in the are encoded close to the BHRF gene, as you can see in the figure. MicroRNA BHRF1, 1, 2, and 3, and then 22 microRNAs are encoded in the BART region of the genome, and that has been shown in the figure also. And microRNAs that are produced in the BART region were usually found to be expressed in latently infected B cells, but when these microRNAs were deleted from the genome and infected into a cell, the virus replicated normally, hence suggesting that the microRNA deletion has not I'm sorry, I take that back. Actually, these BART microRNAs were found to be expressed in latently infected B cells, but after deletion, the virus was still infected and was latent in the infection, suggesting that microRNAs are not involved in the latency of the virus. So, in the case of Kaposi sarcoma virus, 12 microRNAs have been discovered so far. 2 microRNAs are present in the K10 and K12 region of the genome, and the rest of the 10 microRNAs are present in the open reading frame ORF71, and deletion of these 10 microRNAs does not inhibit lytic replication, which means the microRNAs are not involved in the lytic replication, but might be rather involved in the latency of the virus. So, and also when the lytic replication was induced, the microRNA production was not enhanced, again suggesting the same role of microRNAs in latency. In the case of HSV 1, all the microRNAs are present in the latency associated transcript region and are expressed only during latency. And in HSV2 it's the same story, but you can see one microRNA, that is MIR H10, that is not present in the LAT region, and this microRNA is found to be expressed in productive replicating virus and not in the latent virus. And most of the alpha herpes virus microRNAs are considered as latency associated microRNAs. Then we will move on to microRNAs that cause lytic replication. In HCMV, 11 microRNAs are present, and all of these 11 microRNAs are present in the coding regions of the genome, and all these genes are expressed in the early and immediate early phase of replication. So basically these microRNAs are also expressed in the early and immediate early phase of replication, and none of these microRNAs has been linked to latency, but there has not been a very successful model for HCMV to do a further and to do a detailed analysis on the rules of microRNAs. Some scientists have performed sequence analysis of microRNAs from different members of the herpes virus family. There was very little sequence similarity, suggesting no evidence for conservation of microRNA sequences. In HSV1 and HSV2, the microRNAs have a very high sequence homology of 85%, but the seed sequence are not conserved. Seed sequences are usually present at the 3' end of the microRNA, and they are 7 nucleotides long, starting from the 2nd nucleotide to the 8th nucleotide. For successful mRNA downregulation, the seed sequence complementarity is required. The authors conclude by saying that the microRNA sequence might not be conserved, but the functions of the microRNAs may be conserved, because each microRNA can bind to the same target but in different sites. So the sequence might not be the same for all the sites. So that's what they mean by that statement. Some of the viral microRNAs that were studied are those of adenoviruses, and adenoviruses have express two microRNAs, VA1 and VA2, and the major functions of these microRNAs are to downregulate innate immunity factor (PKR). But these microRNAs also downregulate the production of other cellular microRNAs by inhibiting dicer, which is, as you know, an endonuclease that is required for maturation of the microRNA from pre-microRNA. And viral infection leads to upregulation and downregulation of several host microRNAs and viral mRNAs, and some of the roles of the microRNAs can be seen in this figure, and You don't have to memorize the entire figure, but it would be good if you know the highlighted portion of the figure. That is, if the viral microRNA has a perfect target, then it binds to the viral genes, and usually viral genes and viral microRNAs are uh, in the form of a complement. So when the microRNA is produced, they can directly target the genes and downregulate the expression of those genes. So some of these viral microRNAs target some viral genes. For example, in the case of polyoma virus, um, the simian virus 40 encodes a microRNA that inhibits the expression of T antigens, which, as we all know, is needed for early replication of the virus. And another example is Epstein-Barr virus, which encodes a microRNA that inhibits BALF5, which is a viral DNA polymerase, and inhibition of this results in inhibition of viral replication. And the viral microRNAs can also inhibit the expression of mRNAs by an imperfect target. So imperfect target can be either the viral genes themselves or the cellular genes. In the case of viral genes, some of the functions are activation of the lytic and latent switches, and in case and also the growth of the virus themselves. In the case of cellular genes, uh, the microRNAs promote the growth and survival of the host cell, so that the virus can replicate easily, and also they inactivate the immune response against the virus. And these are some of the roles of viral microRNAs on the host genes as well as viral genes themselves. And antiviral immunity and RNA interference. There has not been any report about any microRNAs that has been encoded by an RNA virus, given that if that's the case, then RNA virus has to have a certain mechanism to evade the host microRNA defense, because if you have a mature microRNA that can target an mRNA or a viral RNA, then the replication or translation would not proceed, and the virus will normally be put in a latency stage. But this does not happen all the time. In some viruses, the microRNA binds. In some cases, the microRNA binds to the viral RNA and puts them in latency. But in other cases, such as in HIV-1, this does not occur. HIV-1 genome has several targets for host microRNAs to bind, and the need for these targets is not known, because HIV-1 is rarely latent, and latency is usually seen only in the HIV infected individuals who undergo HART, which is a highly active antiretroviral therapy that puts the virus in a state of latency, and once this therapy is stopped, the virus shoots back up and can be more aggressive in infection. So, what what are the roles of the microRNAs, microRNA targets in the genome? And then HIV has also found to be targeted by a microRNA called microRNA 29A, which is a cellular microRNA. And what it does is it takes up the viral genome, takes the viral genome into the P bodies, and these P bodies are then used by HIV-1 to replicate and stay away from the translational machinery in the of the cytoplasm. So this microRNA and P bodies are basically exploited by the virus to promote its replication and assembly, and therefore help itself to replicate freely. Current research in the areas of viral antiviral therapeutics is antiviral defense by RNA interference. So scientists are using several microRNAs and short interfering RNAs to knock down viral genes and to uh, inhibit the replication of pathogenic viruses. Thank you so much for listening.